Assalamualaikum and a good day to our parents and friends. Saya NURUL SHAHEIDA binti Abdul Aziz. Today I present to you a review of Anticancer discovery from Antarctic animals. A big thanks to my supervisor Dr. Sharifah Aminah Said Muhammad, who gave a lot of guidance in completing my FYP1. First, let's begin with the introduction, which consists of the background of the study, problem statement, research question, objective of the study and significance of the study. The Antarctic is the farthest south on the planet and this continent are coldest, dry, windy and most hostile. The Antarctic is home to a variety of animal species such as penguin, seals, krill whales, nudibranch, several other bird species, various types of seabirds and marine invertebrates. And Antarctic animals are those to be suitable for the discovery of new anti-cancer treatment because of their severe habitat, bioactive chemical and oxidative stress adaptations. New cancer management and prevention techniques are being developed because of ongoing anti-cancer research for the Antarctic. Cancer remains a significant global health challenge with millions of new cases reported annually over the world. Many Antarctic animal species remain fully studied, making it challenging to identify and isolate bioactive compounds from these organisms due to limited knowledge and research of their biology and physiology. It is difficult for scientists to study and gather samples from this organism because of the harsh and remote Antarctic environment. The purpose of this review is to identify the bioactive compound in the kinds of animals for the Antarctic to produce anti-cancer and harness the therapeutic potential as a cancer therapy. This literature review has 3 research questions, with the first, what type of animals are found in the Antarctic to produce anti-cancer? Second, how do the anti-cancer mechanism found in animals in the Antarctic prevent cancer? Third, what type of anti-cancer drug has been discovered in Antarctic animals? The objectives of the study are to discuss the type of animals found in the Antarctic environment to produce anti-cancer. Second, to report on the anti-cancer mechanism of compounds discovered from Antarctic animals. Third, to review the anti-cancer drug that has been discovered in Antarctic animals. But this semester, I only focus on the first objectives and the rest I continue in my FYP2. Antarctic animals have evolved unique biochemical adaptation to survive in extreme conditions like freezing temperature, high salinity and low oxygen levels. Understanding the type of animal and their adaptation in the Antarctic could lead to the discovery of bioactive metabolites that exhibit anti-cancer properties. Some Antarctic animal species possess natural defense against environmental stressors, which might translate into mechanisms that combat cancer. Investigating this mechanism could open insight into how cancer cells might be targeted or inhibited. Next, I proceed about the literature review, which consists of a literature review of background, type of animal from the Antarctic to produce anti-cancer, and the 3 setting research questions. There are many species on the list of Antarctic animals in different habitats that can be found all over the Antarctic, such as the wandering albatross, canto penguin, southern elephant seal, Antarctic crayfish, crevalle seal, emperor penguin, blue whale, kite and leopard seal, and marine invertebrates. But in this literature review, I focus on animals that can produce anti-cancer, which are emerald rockfish, spontaneous, bryozoan, nudibranch, and sea animals. The first animal that can produce anti-cancer are emerald rockfish, which is the Bernache. These animals are important dominant species in Southern Ocean marine ecosystem. This fish has developed specific protein adaptation in their bodies that keep their blood from freezing, which allow them to withstand the frigid water of the Antarctic. Next, the marine invertebrates can produce anti-cancer, can call as spunk, which is K. variosa. The Antarctic spunk K. variosa forms part of the unique spunk community found in the Southern Ocean around Antarctic. To survive in below freezing, this spunk produce antifreeze proteins that stop ice crystals from forming inside the cell. The next spunk is L. apicalis. L. apicalis is one of the spunk species that are essential to the benthic ecosystem in the Antarctic marine environment. They can survive in below freezing temperature because they make antifreeze protein which stop ice crystal from forming inside the cell. Next, for the tunicates, Ephalcladicum is found in a variety of habitats, including deeper waters and shallow coastal regions. As a feeders, Ephalcladicum helps the ecosystem nutrient cycle by drowning in salt water and removing organic particles and microscopic organisms with their unique structure to adapt in this environment. Various one which are bibugula colonies contribute to the overall biodiversity of the Antarctic marine ecosystem by providing other organisms with a substrate and a place to live. As fetal feeders, bibugula contributes to the cycling of nutrients by eating organic particles and plankton from the water column. Next, the marine invertebrates that can produce anticancer are nudibranch, we can call as Biaoxani. Biaoxani are found in both deeper and shallow coastal region, and they feed on marine surface like rocks and algae. These nudibranchs have physiological adaptation that allow them to endure the bitterly cold marine environment of the Antarctic. Lastly, sea anemones, we can call as Thelmatactis species. Thelmatactis species are found in both deeper and shallow coastal region in Antarctic. Thelmatactis species have specific compound or protein that help keep them from freezing or help them modify their metabolic process when the temperature drops. The purpose of the first research question is to use scientists' understanding of the diversity of animal species found in the Antarctic to identify a wide range of animals with unique adaptation and metabolite that have anti-cancer properties. This research question focuses on identifying the animal with the potential to produce anti-cancer compounds and contribute to a broader understanding of the region's biodiversity and its potential benefit in producing cancer therapy. The second research question will resolve the query on the mechanism of anticancer from Antarctic animals to treat or inhibit cancer cells. This research question focuses on the mechanism that provide potential targets for anticancer therapies. It helps in identifying specific molecular or cellular pathways that can be manipulated or targeted by drugs to hinder cancer progression or induce cancer cell death. Lastly, the third research question will seek the type of anti-cancer drug that has been discovered in Antarctic animals. The purpose of the research question is to identify the specific drugs or compounds derived from Antarctic animals that exhibit anti-cancer properties and allow for further research and development in the field of oncology. This is all from my FYP1 presentation. Thank you.